There you go. Great. Um, well, thank you, Anne. Well, good afternoon, everyone, and welcome to our July Global Health Tuesday. And for today, our title is Population Divergence and Local Adaptation of Blacklegged Ticks in the United States. I'm Jorge Osorio, and I'm the new director of the UW-Madison Global Health Institute. And with our Global Health Tuesday webinar seminars series, our institute hosts researchers and practitioners from UW-Madison and across the world. And they actually work together with investigators in many different locations and communities and share their experiences um, to provide insights into global health and encourage conversation, help connect local colleagues and locally and globally. And for tonight, we are welcoming a panel led by Sean Schoville. He's an associate professor in the Department of Entomology in the College of Agricultural and Life Sciences. He is also the director of the Center of Ecology and the Environment. Sean received his doctorate degree in environmental science policy and management at the University of California, Berkeley, where he also completed his undergraduate training. And the major areas of research are rapid evolutionary change, biogeography, biological responses to global change, species diversity and conservation, insect physiology, and alpine biology. So tonight, he and his teammates will present the work that is completed with funding from the Global Health Institute Seed Grant. Please join me in welcoming Sean, and he will introduce his panelists. Thank you.
Hi, everybody. Thank you for the nice introduction, and welcome to the webinar tonight. It's a pleasure to be here sharing our work with you. Yes, this was funded by the Global Health Institute of Wisconsin as a seed grant, and it's just a really nice example of the impact of that funding, bringing together a new team of collaborators. So I'd like to introduce my collaborators just briefly. I'm joined today by Dr. Jean Tsao from the Michigan State University, as well as Susan Paskewitz here at UW-Madison and Department of Entomology as well. She's the director of the Midwest Center of Excellence on campus. And then I also have my PhD student, Dahn-young Dong, who's gonna join us as well. Jean, Dahn-young, and I myself will give short talks. Each one of us will share some slides. And then Susan's here as well to help us address questions in the Q&A period towards the end. On that note, what we'd like to do as we go through the talk is ask you to enter questions in the chat box. And at the end, we'll work through the chat box and try to address your questions orally. But given the number of people involved, we might not have audio questions. We might just use the chat to answer questions. So the sequence of events, Jean will lead us off to kind of give us a general background on ticks and their biology. I'll talk more specifically about the population genetics data that we've developed and what we've learned from that. And then Dahn-young will take over, share some of his insights on that data as well, and then kind of lead into our future directions. So without further ado, I'll let Jean get us started here. Thank you.
All right, well, thank you very much for having me participate in this talk, and I'm really glad to be with everyone here on a Tuesday summer afternoon / evening. As Sean said, I'll give us the introduction. So, at the mile high global view, there have been a lot of emerging infectious diseases in the past half century, as most of you know. And so this is a paper from 2004, and you can see here they've noted lots of emerging and/or re-emerging human infectious diseases. And some of the names that are larger, I'm sure you're familiar with. And it kind of harkens back to, not to belittle any of these diseases, but it seems like, oh, that was just so long ago, because in that time where these people talked about how there's been all these anthropogenic factors leading to the rise of emerging infectious diseases and that there will be more, it was true, there were more. And so there was Zika, and then Ebola reemerged, and then MERS, the coronavirus. And of course, everyone is familiar with the SARS-CoV-2 virus leading to COVID that we're still dealing with. And most recently, I'm sure you've been hearing about in the news and reading about, but monkeypox that's been spreading around the world. So most of you may know, but if you don't, many of these emerging diseases, actually most of, depending on which review you look at, you read up to 70% of the emerging infectious diseases of humans have a zoonotic origin. So what does that mean? That means that these pathogens normally, naturally, are maintained by animals. They can be wildlife, like the animals on the left side of the slide. They can be domestic animals, such as the pig and the chicken, livestock that can be transmitted to companion animals, and so these animals can transmit these pathogens to humans directly, and or they can transmit them to what we call vector species, often arthropod vectors, which are insects or arachnids that bite humans. And so, for instance, on the left side, there's the flea, then in the center, a silhouette of a mosquito, and on the right, a tick. And we're mainly going to be talking about ticks today. So in the US, zooming into the US, the incidence of vector-borne diseases has been increasing over at least the past 2 decades. So this paper was published in 2018, and they counted up the numbers of, the incidence, the reported cases of mosquito-borne and tick-borne diseases. And in each year, you see 2 bars. One on the left is due to mosquitoes, on the right is ticks. So you can see there's been an increase over time, and also most of the tall bars are due to ticks. And so they accounted for about 75% of the reported tick-borne, vector-borne diseases to humans. So in the US, there are multiple tick-borne diseases. There are more than the 6 that are listed here, but these are 6 that are reportable. And what's interesting is, so let me back up and say each dot represents one human case that's been reported to the CDC that matches or that meets the epidemiological reporting criteria, right? And what's interesting is that the blacklegged tick, the tick that we're really going to be discussing today, it accounts for at least 90% of all tick-borne diseases. And you can see circumscribed in this, in the navy blue or bright blue dots, that's where the majority of the black-borne tick or deer tick, it's also known as associated diseases, are spread. And so this is the tick. It is very much like other ticks you may have heard of. In Wisconsin, you're probably very familiar with the dog tick, the American dog tick, also known as wood tick. There's also the Lone Star tick. But these ticks that often bite humans, they're what we call 3-host ticks. And so there are 3 life stages post-egg that can bite humans. And so we start off with a larva that's 6-legged over there on the top left, and it hatches for the most part without any infection. And then you get the nymph stage, which is the teenage or adolescent stage, or 8 legs at that point. And that is often the epidemiologically most important stage. And then we have on the right, you now have the sexes morphologically distinguished, and you have the male on the left and the female on the right. And the female can also transmit. And so after the nymph, the female is the most important vector. The male doesn't feed, and so it's not important as a vector. But you can look at the list on top. It's associated with several human diseases, several of which are also known to cause diseases in our companion animals. And these are just the ones, the diseases that we know about. But all of these diseases, Lyme disease is the most important. It is the leading vector-borne disease, and anaplasmosis is the second one. And Lyme disease is purportedly experienced by about 400,000 people a year, and that number is probably increasing. All right. So one of the main reasons why diseases transmitted by the black-legged tick have been increasing is because the distribution of the tick has been increasing. Its population abundance, let's say first, has been increasing, and then of course, then spilling over and spreading. And so the map on the left, in the red counties, those are the counties where when researchers look through the literature and looked through the published literature and the gray literature, they saw that the numbers of ticks reported constituted an established population. And blue counties were just that they were detected, but they didn't meet the criteria. If you look at the map on the right, which was just an update of that map on the left, with, by 2015, you see many more red counties, all right? And now I'll show you in the future slides, there are even more red counties. And so these tick-borne diseases have spread because the tick population has grown and spread, and also because we really don't have measures that can effectively reduce the environmental risk of contacting these ticks, or human vaccines that can reduce our risk of contracting these diseases as well. And so, although there are some methods, such as the ones on the right, culling deer and using acaricide, that have been known to be effective at reducing tick populations, number one, often they are not everyone wants to use these methods politically, socially, economically, as an area-wide public health prevention method. They just haven't caught on, let's say. And number two, there's some evidence that shows that at least at the levels at which these practices are practiced, we'll just say, they actually may not be as effective as we would like to reduce effectively the numbers of people getting contracting these diseases. So that is what a lot of researchers now are working on, and what the Midwest Center of Excellence hopes to be working on more in the next few years to improve many of these many methods to really reduce tick numbers in the environment and contacts with people. So right now, then, the only way we really have to reduce tick-borne diseases is really through education and telling people, please adopt these measures that have been shown to be effective at reducing your risk of contacting ticks and therefore exposing yourself to disease. So it's knowing where ticks are found, when they're found, what kinds of activities are considered higher risk. It's taking prevention measures such as using an EPA registered repellent. All right. And also knowing where to look on your body and to know that you should check often, right? So a lot of this then depends on public health being able to tell people where there are ticks, where there are infected ticks, and when there are. And so that's what a lot of folks like myself in the past have really worked on the ecology of the ticks and trying to understand their distribution and abundance and the factors that have been promoting their spread. And so, for instance, and I think this picture on the left is from University of Wisconsin researchers, but we know that changes in the landscape due to, again, driven by anthropogenic desires and causes, have changed the landscape such that it's improved the land, the habitat for ticks, as well as, really, what's very important, the habitat for the wildlife that the ticks need to feed on. So we create more habitat, that's so that the ticks have a place to live, and they also have food, right? And then, as you can see from that picture, it's the way we've changed it is oftentimes we've made subdivisions or areas where people are recreating. And so not only do you have the ticks and the wildlife maintaining cycles of disease, you bring the people in, right? And so that's why it's a problem. If people weren't there, we wouldn't care as much, right? At least for a zoonotic disease. And then also changes in climate has also facilitated the survivorship and the spread of this tick, but the effects of climate, although we often think about it and wonder how important it is, probably really varies. And so areas in the northern edge of the range, that's probably where climate change has helped promote the tick population. May have been facilitating somewhat in other areas, like in Wisconsin, Minnesota, Michigan, in potentially incrementally increasing survivorship rates, host finding rates, but they would be here, and it may be just the rate of spread is potentially higher. But these ticks are also moving southwards, and in those areas, at least in the mid-US, climate was fine, right? But potentially on the southern edge, climate might actually change. Those changes might actually make it worse for these ticks. That's to be seen. So I've kind of talked about the factors of the ecology that might contribute to the distribution and abundance and spread of these ticks. Our labs have been working on this, all right? And our labs, and mainly Susan's lab, and I hope to be doing more of, have also, folks in our lab have also worked on methods to try to reduce ticks and the risk of exposure to people, such as, doing changes in the landscape, removing some invasive species that are known to create some of that nice habitat for the ticks and their hosts. Targeted use of acaricides at the right time of year, and places in the yard to try to reduce tick populations, or numbers, I should say. And then also host targeted methods. So instead of spraying acaricide more broadly, target the hosts that actually feed the ticks, right? So now those are some of the methods, so people are doing ecological research to understand where ticks are, where they're moving, what might limit their distribution. But actually these ticks are generalist parasites, both in terms of what they feed on and where they live. So, it's kind of hard to get rid of them in that sense, but also methods to try to target them and to intervene in the cycles to reduce their population numbers. Now I'm going to leave you with two patterns and then a little bit more about the biology, so that then you'll have more context to understand the rest of the talk. But here, just zooming in, we've talked about how the ticks have spread in the eastern US. But here, just in the upper Midwest, you can see more clearly the numbers, percentages of red counties, again, representing established populations, have been increasing. And we expect that still to continue. Now, Minnesota and Wisconsin are almost full. Michigan is getting there, but there are still some other areas that they're spreading, right? Here, zooming back out to the scale of the eastern US, you also see that the numbers of red counties have been increasing, right? And as I said earlier, they are spreading southward, right? So basically a tick will go in any direction until it hits water, or perhaps in the central Midwest, or really the West, if it gets too dry, all right? But it's interesting, as it moves south, all right, it is important to note that the ticks have been found in the South. They are established in some areas in the South, and they're probably, as the yellow counties show, they're probably in some of these other areas as well, or they will be. The issue is, look over on the right, the case, to remind you, the cases of Lyme disease, and this reflects the other black-legged tick-associated diseases as well, but they are concentrated in the north, right? So just remember that, and Sean will probably talk more about that. All right, so then just to have some more biology and context in your mind, this tick, the same as the American dog tick and the Lone Star tick, but differently from mosquitoes, it only takes one blood meal, full blood meal per life stage. So they need that blood meal to advance from larvae to nymphs and from nymphs to adults. And the female needs that blood meal in order to lay eggs, all right? But that blood meal is what, how pathogens get transmitted. These ticks, as I mentioned before, are generalist parasites. So they can feed on mammals, songbirds, and lizards if they're around. They use an ambush strategy, so they don't fly around. So that's important to know for thinking about gene flow and all that, right? So they don't fly around, but wherever their hosts go, they can go with them. So you can think of that in the scale of a mouse or a deer or a migrating bird, all right? And microhabitat in terms of local survivorship is very important because they're very desiccation prone, and the habitat can also buffer them from extreme temperatures. And so with that little primer, I'll pass this on. I'll stop sharing, pass this on to Sean.
All right, I'll get mine going here. All right, thank you, Jean. That was a very nice introduction. So what our project really focuses on is employing modern genomic sequencing techniques in order to illuminate how well the ticks move in their environment, and also if we can find evidence of them adapting locally to different environments across the range. On the right, you see a figure of their life cycle. And Jean talked about how they use these different animals to host on for blood meals. Well, one factor that's quite interesting in that interaction is that the hosts that are available change geographically. So in the north, we have lots of deer. In the south, there's a variety of lizards and so forth. And so those hosts change as you go to different geographical regions. And the ticks are thought to maybe adapt to those different hosts in the different parts of the range. So one of the things we'll look at in particular with the genomics data is whether we see evidence of them differentially adapting to hosts. And then just more in general, we want to understand how they're moving in their environment, and can we use the genetic data to improve our understanding of how readily they move across the landscape. So an overview of our data set is as follows. The map shows with yellow dots where we sampled ticks. We sampled from 9 states. In the states of Wisconsin and Michigan, we sampled at many sites because we had ready access to those samples. The data set includes about 92 ticks. That doesn't seem like a large data set. We actually have thousands of ticks at our disposal. The reason we are limited to that size, that number of ticks, is because it's quite expensive to use genome sequencing. So you can see my cost estimate there for every sample. It's about $235 to get its whole genome. That's in part because the technology is expensive, and also the tick genome is very large. It's almost as large as a human genome, so it's a very complex genome, and it takes a lot of money to sequence through that and provide reliable data. So this is a distribution or a sampling map. Before I get into some of the results that we have, I do want to give you a little bit of a background on some of the work that's preceded us. So ticks have been of interest for a long time, of course, and they were some of the earliest insects and arthropods to be studied with genetics techniques. So there's some papers from the early 90s that started looking at ticks and their genetics. And so I've kind of listed those papers here to acknowledge them, and also to summarize some of these earlier findings. The picture on the right on the top, Ixodes scapularis, that's our black-legged tick. The bottom panel is Borrelia burgdorferi, the causative agent of Lyme disease. And what the picture shows is part of a genome. This is the mitochondrial genome of the tick, and variation in that genome. The variation is indicated by the little branches that you see emanating between the circles and the squares. So each step is a different mutation, a change at the DNA level. And with this early data from the mitochondrial genome, what was found is that there was differences between the North and the South in terms of the genetic variation they had. Indicated in black and gray is the northern populations in the Midwest and Northeast. And you can see there's only a few of these alleles represented by the squares and the circles that are represented in that region. Whereas in the South, there's a lot more diversity and a lot of different alleles that were represented. Another thing that was found for the black-legged tick is that the levels of variation that distinguish the North and the South suggested that they were separated for some time, and that the North was probably expanding population. It looked like it had grown rapidly over time, most likely from a response to the glacial cycles. So you'll see that again, and as I go forward in our results, some of those same patterns hold. And then finally, there was analyses of Borrelia burgdorferi, this Lyme disease agent. And what was found is that their history is not a mirror image of the black-legged ticks themselves, suggesting that they have an independent evolutionary history, and they have variation that's often more refined on a local landscape scale. So there's differences between Midwestern strains of Borrelia and Northeastern strains and so forth. And you can see that geographical variation if you look at the genetic data. In addition to some of these early studies, a series of studies followed, and they expanded the scope of sampling geographically, as well as in sample size, and then also looking at different parts of the genomes of the ticks. So that earlier work looked at a mitochondrial gene. The mitochondria is an organelle in every cell, and it has special properties. It's maternally inherited. It's got it's haploid, it's a single copy. So it's very easy to study, but it's a very limited snapshot of the genetic diversity in these organisms. So as time went on, people added more data. They started to take data from the nuclear genome, which has a lot more information about the history of the tick. And what this expansion of data has shown is that the story is a little bit more complicated. In this plot here, you can see on the left a map of the sampling locations. And then on the right, there's a bar chart. And I just want to draw your attention to, let me see if I can get my, well, oops. Just to the far right where it says k equals 4. And that bar lines up with the geographical map on the left. And basically the genetic data suggests there's at least 4 distinct genetic populations that occur along the East Coast. There's a group in the north. And then as you get towards Virginia and south of there, you see much more broken up structure. So there's different populations that are evolving somewhat independently. What this data showed is that there may be more limited dispersal of ticks across these regions. And in particular, the South seemed to be a little bit more structured than the North. Another study, and this dates to 2015, also looked at nuclear genetic variation in the tick. And they try to use this data to estimate rates of gene flow. That is the amount of genes that are exchanged by migrating individuals through time, so that variation that's found in one population could contribute to the genetic variation in another population. And these maps sort of show you a series of arrows between sites that were studied. The width of the arrow indicates the strength of inferred gene flow, and then the direction of the arrow is indicated by the pointer of the arrow. And what they generally found is that in the northern region, there seemed to be high levels of gene flow among sites. In the southern region, there also seemed to be high levels of gene flow. But when you looked between the north and the south, it seemed to be more limited. And in fact, they inferred that gene flow most likely occurred from the north to the south in an asymmetric fashion, so in a unidirectional fashion. And then finally, in 2015, the tick genome was sequenced. It was a large effort. A big consortium of scientists were involved in developing this resource. And you can see some details about the genome there. It's estimated to be about 2 gigabases. This has been revised as we've improved our sequencing over time. What this study also did was take a sample of series of populations and look at genetic diversity now with a little bit larger data set. So here we have a set of 30,000 markers scattered around the nuclear genome that they use to try to infer genetic population structure. And the bar chart shows you some of those populations. If they share the same color, they're thought to be within the same population. And what you can see is that there's a group of populations in Florida, North Carolina, that are distinct from everything in the north. But this had a strange result in that it found populations in Wisconsin, Maine, and Massachusetts formed one population, while Indiana and New Hampshire formed another. If you look at the map on the left, the locations of those sites, that doesn't make a lot of sense in how these different populations that are in the north would be separated when they're geographically overlapping. So this is a bit of a background on what was known to date. So now we enter into our data set. The data set is quite large. As I said, it's the whole genome. That represents about 22 million different markers throughout the nuclear genome. So it surpasses any of the previous data sets in terms of its size. And this plot here kind of gives you a general picture of the genetic variation we found across different populations. So this is showing you a statistic we call FST. Basically, that measures differences between population pairs while controlling for variation within those populations, standardizing by that variation within populations. And it's hard to look at these sort of tables. So I'll just kind of generally explain what's going on. If you intersect 2 of the points on the table and look at the column or the row headers, you can see what population pairs are being compared. And I've color-coded them. So if they're color-coded blue or yellow, that means there's very little genetic difference between them. Their numbers, the FST number, as it approaches 0, means there's no difference. And as FST goes towards 1, it means very strong difference or complete differentiation. So in the upper corner of the table, comparisons among Wisconsin, Michigan, Maine show that there's very little genetic differentiation among those populations. The populations in the north are very similar to one another. We also see in blue groups in the southern populations, blue and yellow. So Virginia to Florida has very little genetic differentiation among those populations. What's interesting here is in red, we see strong differences that emerge when we compare anything in the north to anything in the south. So there really are very strong differences between northern populations and southern populations. FST values on this order, 0.37 or so, are very high and suggest that there's been long-term isolation. Effectively, there's no gene flow likely to be occurring between these two regions. There's also an interesting note here, and it's shown in orange, that Tennessee is somewhat different. Tennessee is a little bit more similar to the north than any other southern population. It's also a little bit more different from other southern populations. And so something interesting is happening in Tennessee. There might be yet another distinct region of variation for the ticks. This is another way to present this same genomic data. And on the right, you see a plot. It's multidimensional scaling, but it's a way of summarizing the genetic variation among all the individuals sampled, and then plotting them based on their genetic similarity to one another. And you can see there are 3 clouds of points here, and I've labeled them according to their regions. So we see this group of the Midwestern and Northeastern tick populations that form a clear genetic grouping. We see the Southeastern US forms another genetic grouping. And then Tennessee is off on its own, somewhat different from both of those. So this sort of recapitulates the FST measures that I just presented, provides a different view of how to look at that. But it does show that we have, at this large scale of the US, strong population genetic structure, and most likely limited gene flow across these different regions. One of the things that we've added in our analyses that wasn't looked at carefully before in some of those early papers is an analysis of how genetic variation changes across distance. So you can see here on a panel on the lower left is a regression. The regression is of genetic differentiation among populations. That's the FST statistic. And then the x-axis shows geographical distance on a log scale. So kilometers of distance on a log scale. And you can see there's a relationship there. There's the line that's drawn is a positively increasing genetic differentiation as you go across distance. And that's a significant regression line, statistically significant. And what that tells us is that there's gradually over space, genetic differentiation accumulating among tick populations, meaning that they're dispersal limited. So we don't have ticks from the south sharing genetic information with ticks in the north, and even more so on a smaller scale, probably at the scale of states, hundreds of kilometers. There's limits to the movement of ticks so that genetic differences arise over these much more shallow scales at the level of states. This result is also important for helping us understand why some of those earlier studies seem to disagree on directions of gene flow, the number of populations that were found. It's because some of the techniques that were used are sensitive to this pattern of isolation by distance, and the authors overlooked this factor during their analysis. And so with this knowledge, we can revise our understanding of sort of the population structure of ticks more reliably. Okay, in addition to just sort of understanding the overall genetic patterns, we can use the whole genome data to reconstruct population size changes through time. So this is a plot. This shows patterns just in Wisconsin. We've done this for each of the populations that are under study. And the way you look at this plot is on the y-axis, it shows you estimates of the numbers of breeding individuals in the population through time. So these are individuals that contributed to the next generation's genetic variation. We call this the effective size. And then on the y-axis, there's time. Time here is in thousands of years on the bottom x-axis. And we sort of go from the left to the right of the graph to look at how population size changed through time into the past. The red line is the estimated median value of the population size through time. And then the little gray colored bars are different confidence intervals around that estimate. This method is probably not very reliable for recent time, but it works very well once we get back to thousands of years ago. And what you can see here in the plot is there's a big dip in the kind of right of the graph that I pointed out with an arrow that shows a strong population contraction in the northern and in this Wisconsin population. This coincides time-wise with the last glacial maximum. I'll show you a plot of that geographically in just a minute. But so we can see the impact of some of these longer-term climatic events on the genetic diversity and the evolution of these populations through time. What's interesting is that only the northern populations show this response to glacial cycles. In the south, patterns of effective population size change are more variable. Some populations seem to be very constant across time. Others seem to fluctuate, but not necessarily in relationship to glacial cycles. Probably some other regional climatic events were affecting their populations. So this is just that map of the last glacial maximum. Shown in blue here is the extent of the Laurentide ice sheet that came into the Midwest and the East Coast. And you can see the timing of that here. And you can see that's just the distribution of the ice across the different states. So based on our data, it makes sense to think that the Wisconsin, Michigan, Massachusetts ticks, all these ticks in the north, would have had to have been distributed south of that ice sheet. They couldn't have survived under ice. And then they would have recolonized these habitats starting, anywhere from 20,000 to 15,000 years ago as that ice receded. So that helps explain the genetic similarity among some of these ticks in the north, and also explains some of the diversity patterns that they have. A little bit less diversity than the ticks we see in the south, and then they have this history of population size change through time. Probably the most interesting part of our analysis focuses on evidence for selection in the genome. And I'm just going to share a couple of highlights from that analysis without going into the methods. On the table here, I've shown a set of genes that we've identified to be likely under selection. And these are differences that would occur across populations. So they indicate local adaptation in the tick populations. And I'm just showing you some of the top 20 genes that were shared between two different tests. So they're very strong evidence of selection. And I've color-coded some of their functions here just to talk about some of the results. So what's interesting in this set of genes is that we have genes that likely play a role in host interactions with during blood feeding. So genes that play a role in agglutination of blood, in cell adhesion, so the way cells bind together. These metalloendoproteases and peptidases can attack, metal-containing compounds, so they help break down blood. And so these suite of genes suggest that there's likely differences in the way ticks respond to their local host. So these are critical genes that are expressed in their saliva during blood feeding, and they've evolved over time to become more effective in the different populations they occur in to the host that they're exposed to. So there are these differences between tick populations in terms of their abilities to feed on different hosts. I also highlighted in green these G protein coupled receptors. These are kind of interesting for a number of respects in the tick biology, but the reason I draw attention to them in the talk is that our next generation of pesticides one of the candidates for targeting ticks, it's been suggested that we focus on these G protein coupled receptors. Typically, pesticides target the nervous system in some way, and these are kind of receptors that help in cell signaling, and so are thought to be good candidates for novel insecticides. Well, what we've shown here is that there's actually differences among tick populations of these genes, which may throw a bit of a wrench into any plans to develop really effective insecticides targeting them. If there's a lot of variation, insecticide may not work reliably across these different populations. So a second kind of summary view of some of the genes under selection is this slide here. So there were a lot of genes that we found under selection. It's difficult to talk about them all. But what we can do is we can look for certain functions that many of them share and see if they're overrepresented based on what we would expect. And so I've shown you here some of the biological functions that these suites of selected genes are involved in. And you can see that highlighted in bold, there's a whole suite of genes, in this case, 34 genes, a whole suite of genes that may be involved in host defenses. And there are other genes like the heme binding genes and the monooxygenase activity-based genes that are also probably involved in host feeding. They may interact to detoxify host defenses. They may also, help the ticks sort of break down the blood proteins and so forth that they're feeding on. And then finally, there's a signal of evolving to the environment that jumps out in response to light stimulus and external stimulus. That's most likely associated with photoperiod variation as you go from southern states to northern states. So the ticks have to change their life history, their activity time based on the seasons. And so the sensitivity to these light stimuli and so forth might be under selection. So this work fits into a broader context, and that is that a number of studies have come before that suggest that there are differences between northern and southern populations, not just in their genetics, but also in the way they interact with their hosts and their behavior in their environment. So some of the things that have been studied are what we call the questing behavior. That's how ticks choose to pursue their hosts for blood meals and sort of their activity time and duration. We see host variation. So notably, there's not many lizards in the north, but there's plenty of them in the south. So that's a big driver of how they adapt to their local environment. And then a variety of other abiotic cues, including those light sensing cues that I mentioned. So what's interesting, Jean has done this work with some colleagues through the past, is that there's quite a difference in the behavioral ecology of ticks as you go from south to north. And one of the ways that that's been measured is in the host-seeking behavior of the ticks in the nymphal stage. And what we see is that they're much more active, much more prone to quest and actively search for hosts in the north than they are in the south. And so this sort of shows you a pattern of a density of those ticks, but also on this slide, we sort of measure the behavioral response, how often they search out for blood meals, it's a probability of questing, versus the incidence of transmission of Lyme disease at those locations. And there's a strong relationship with Lyme disease being more prevalent in the north and there being higher questing activity in the ticks, so behavioral activity that leads to transmission than there is in the south. So our genetic data may suggest that there are differences in ability to use different hosts, as well as maybe even host preferences between these populations in northern and southern US. Okay, I'd like to transition and let Dahn-young share a few slides to continue this work.
Great. Thanks, Sean and Jean. Well, so in the past 30 minutes, we've been talking about tick biology, ecology, and also looking into the genome of the neutral variation population structure, and also how ticks different populations been adapting differently, and there are various genes that have been selected in different ways in different geography. We have this whole genome sequenced, and we're also curious about how ticks are interacting with the biotic environments and with the communities, especially the microbiomes. And using the whole genome sequence, we're able to probe that a little bit more in depth, so that we had a microbiome analysis using Kraken and Bracken. So we used the gene sequence identity, and we ID'd the microbes down to species level, and also used Bracken to estimate relative abundance of each microbe. So here's a heat map where we can see the relative abundance. And in this case, it's on a log scale. So the more bright color is more abundance of particular microspecies, and more cooled color means more lower in abundance. And if you look at the heat map, the vertical lines are the individual tick samples, and the horizontal lines are the species identified. And these ticks are also sorted geographically. You can see the metadata on top, where it's divided by northern regions and southern regions. And since we know that Tennessee, it's sort of independent, kind of different from the rest of the three regions, we marked it at the end here in gray. And also in more detail, we subdivided the states into, the regions into states. The first thing that draws our attention is the presence of Borrelia burgdorferi. We mentioned the causative agent of Lyme disease, and it's very much exclusively limited to northern regions. It's not surprising. We already know that it's probably due to limited gene flow of ticks where the hosts are not carrying the ticks around. And also, Jean and her collaborators have been working on this questiness, the behavioral differences in the north region, south region, and also knowing that in the south we have a very different selection of hosts that are very not efficient in passing on this disease to tick. So that's all the reasons we can see that Borrelia burgdorferi is really present in different ways across north and south. Secondly, we see this heat map is majorly populated by Rickettsia genus. And I would like to draw attention to Rickettsia buchneri, which is an endosymbiont. It's been with tick evolutionarily throughout the time, probably ancient enough into the glacial maximum, where it's present for all the ticks, regardless where we sample them. And we know that it's essential for the nutrients, the vitamins that ticks take, because of its blood feeding lifestyle. It lacks some essential vitamins that this endosymbiont can provide. And interestingly, I didn't think about Rickettsia felis at all until it showed up in high abundance in North and South Carolina. It could be an artifact, but I looked into literature. It's been showing that it could be potentially a co-feeding mechanism with cat fleas that somehow uniquely happened more often in higher abundance in North and South Carolina. But for sure, there are many other Rickettsia species that are just essentially probably imprecise measurements due to this platform, and also given that whole genome sequence, it's large and might be able to capture a lot of the nuances where the genomic divergence of Rickettsia genus is too limited to be able to, really identify the correct species. And lastly, we also identified some other minor pathogens that are not highly abundant across the regions, but we did see Acinetobacter species, Borrelia miyamotoi, which causes relapsing fever, and also Anaplasma phagocytophilum, which causes anaplasmosis, as Jean mentioned at the start of the presentation. And they are not abundant across regions, but they're isolated incidences across the region. So there's no a clear geographic difference in that case. Lastly, since we did this whole genome sequencing, we did not intentionally clean out the surfaces of the ticks. So we were able to pick out some environmental contaminants, but literature has been saying we just needs to read the data with caution, because a lot of times we're not really able to identify whether these environmental sourced microbes are having an impact on the tick evolution or not, or simply some nuanced detail with it, or is simply a artifact that we should ignore. Well, so as we mentioned earlier, we're really interested in the rapid invasion of ticks into the south, and also going up north into Canada. So we're super interested in how this invasion happens and which direction it's moving into. And for future research, we'll look into landscape genomics as a way to really understand the rapid expansion is happening right now. And because we've been looking into the selection and local adaptation patterns, we're also curious in how we can or not effectively design acaricides to be able to manage tick populations and cool down the population density. And lastly, given the climate change, the region's been changing temperature unpredictably, and sometimes in large scale, we'll be able to hopefully predict where the tick's been going to and whether there will be an increase in Lyme disease risk. With that, we'd like to thank Global Health Institute has been providing us generous support, and also many other funders around the region. And thank you also for our collaborators who are in Michigan and also at Wisconsin for helping us with this research. And with that, I would like to take questions, and I'll hand it back to Sean.
Great, thanks, Dahn-young. Thank you all for joining us and sharing this talk with us. What we'd like to do is field questions, and if you would like to just put it in the chat box, that'd be great. We can read them, and Susan will join us for this and help us answer some of these questions. So if you have questions about mitigation strategies as well, we didn't really talk too much about that, but Susan is an expert on some of those strategies here in Wisconsin. We'd be happy just to field any sorts of questions you have.
Sean, what's your best guess for what's going on with the Tennessee ticks?
Yeah, as it happens, Tennessee is just to the west of the Appalachian Mountains. So my immediate thought is that movement of ticks in the south is somehow impacted by that biogeographical barrier. Historically, they've moved along the East Coast, but as you go west from the East Coast, those Appalachian Mountains limit their movement somehow. Whether that's via their hosts or what, I'm not sure, but that would be my first assumption. But they're somewhat a little more similar to the south than they are to the north. So they've been, in contact over evolutionary time with those southern populations, but they really are still quite different from north. I see Jonathan as a question. Go ahead, Jonathan.
Yeah, easier to ask than to type it.
Sure.
Speaking of these southern ticks, I have a question regarding the change in behavior, like alternative questing versus what Dahn-young mentioned about the reservoir competency differences amongst the host. And my understanding, and this is from Rick Osfeld's work, is that lizards and salamanders are not able to carry the spirochete, the bacteria. And so from your presentation, I'm left wondering, are there less cases of Lyme disease in the south because of the lack of, because the ticks are feeding on more of these reservoirs that don't carry the disease, or some sort of questing behavior, or both?
Can I jump in? I think both. I mean, both are factors that play a role. But what I often say is that even if the ticks were 100% infected, that because of this difference in questing behavior, you still have very little Lyme disease in the south. But I think it's entwined. We don't know exactly why they have this different questing behavior. Is it ultimately driven by host selection? Is it due to because the lizards somehow are in the habitat more and they're more likely to be successful at completing their life cycle because they feed on lizards versus small mammals? Or is it because the abiotic factors are such that their survivorship is increased more when they're below the leaf litter more, and that somehow places them more also in contact with lizards and specifically skinks than on some other animals. They aren't that much on on fence lizards. They're not so much on snakes and such. but these skinks are clobbered. So probably both, but I don't know, ultimately versus proximately. If I could ask a question too, maybe some others have some of these questions. I was just curious, these values of FST and such, I mean, how do they compare, I guess, when you say that maybe to some other organisms and such, that to say that they are very, not much gene flow relative to, and how much variation, since it's a ratio too, what is the amount of, I know you sampled single ticks in, areas, but how much variation is there, I guess, within, population? Yeah.
Yeah, it's a good question. FST is one of these tricky statistics that's hard to compare across different species. And we don't often do that for a reason. But I'll say that the levels that are distinguishing the North and the South are very large in general, to the extent that you might see those between, different subspecies, different, and there's a bit in the literature that these were once called different species, the South and North. I think there's a basis for that genetically, though I wouldn't argue that they should be a different species. I just say that they've been isolated for a long time, and they're probably not exchanging genes at present.
Mm-hmm.
But there are probably interesting places. One of the faults of our study is the limited sample size and the gaps, the geographic gaps, right? So we have Tennessee and then up all the way to Wisconsin. There's nothing there, nothing sampled, and there's certainly ticks there. Similarly, on the East Coast, there's this interesting transition between our sample in Virginia and then suddenly this northern population. What's going on, as we move through Virginia and go a little further north? Maybe these two clades mix. So those would be areas where we'd want to take a close look at that. And I see Jonathan has another question.
I don't mean to ask a second question, but I don't see other hands up. So here's a question related to a parallel with climate change. We look at the future and we say, oh, Wisconsin will be like Arkansas is now, or, and it'll be shifted. So with such geographic variability in these ticks, do you think that, 20 years from now, we'll see, Tennessee behaving ticks will be in Wisconsin, and Wisconsin behaving ticks will be up in Alberta somewhere, or at least, or in Ontario anyway?
My thought, given the historical patterns of genetic divergence, is that they don't move effectively over large scales. So I would think that southern populations would lose out and simply decline, if the climate becomes unfavorable for them down there. They wouldn't effectively shift and move to the north. Now, the caveat there is, as I mentioned, Virginia and it's sort of these places in the east where some of these southern lineages may be mixing already, and maybe they'll be able to move up. There's enough gene exchange that their genetic variation will move north. But I wouldn't expect that too much. I see there's a question from Eve in the chat, and I don't know if Susan, you've seen that, but maybe you could comment. The question is about Anaplasma and why it's so rare in our data set. Well, remember, we only sampled 92 ticks, so we have a very small data set for assessing levels of prevalence of the pathogens. But maybe Susan, you could comment on how common Anaplasma is in Wisconsin particularly.
Yeah, in human cases, the reports to our Department of Health Services are usually between 500, sometimes as high as 700. So it's, the second most common tick-borne disease in the state. And I'd also say, I don't know how good the reporting networks are. There's been a lot of talk about the limitations of reporting. And for Lyme disease, people generally seem to think that it's maybe tenfold under reporting. So whatever case numbers we have is significantly off. We've done a lot of sampling of ticks and a lot of looking for Anaplasma in those ticks, and it varies a lot from location to location. We've had some places where as many as, 16% of the nymphs are infected, and then other places where it's closer to 5 to 7%. So there's a lot of variation. It doesn't usually get down into that 2 to 3% though, pretty much anywhere we've looked in the state. So it's common. I think I would have expected to see a little bit more of it in the ticks, the 92 ticks. Those 92 ticks from Wisconsin, or those are 90? Yeah, right. So it's not that many from Wisconsin.
Yeah,
so,
about 30 from Wisconsin. So
Yeah. So I think what you found is probably dead on.
Or 20 from Wisconsin. Sorry.
Close. I don't want to ask a question if no one else is, but I did have one for Dahn-young. Did it look to you like gene samples from Michigan were different in the pathogen or in the microbe profile? It seemed to me there were a couple things that were specific to that sample.
I would say that Wisconsin and Michigan, in terms of the heat map, are quite similar. And there's no broad scale sort of differences from what I can see. I was like a thought that since Sean said there is genetic structure across those, and but it seems that the microbes don't sort of follow that genomic divergence of ticks.
You didn't catch when we spit in there, some human pathogen ticks.
Well, there is this Cutibacterium acne, which could be a contamination, but I won't comment more on that.
We have reached 5:30, so if there aren't any more questions, I would like to really thank all of you for your information and for keeping track of these ticks that seem to be catching up to us. Thank you very much.
I think we all enjoyed that. So thank you for the opportunity, and best wishes to everybody. Stay safe this summer and stay tick free.
And we will share the video.
Thanks.
Thank you.